Hello, this is part 3 of a 3-part mini-lecture series on plant form and function. In part 3, I cover plant growth. The focus of this chapter is really about the form and the function of plants, particularly plant structure and how it lays the foundation for plant function. In this part, we're going to talk about primary growth, which extends the plant body, and secondary growth, which is going to widen shoots and roots. When we talk about primary growth, we're really focused on the different types of meristems. And remember, that's undifferentiated cells that are going to continually divide and reproduce or produce new cells. And we particularly focus on the apical meristems, which are going to be at the tip of each root and shoot, as you can see here up at the top. And those cells divide, enlarge, and differentiate. Root and shoot tips extend plant body outward, allowing it to explore new space, usually for increasing surface area and acquiring water and nutrients. Apical meristems are pictured here. You can see the apical meristem that's located in the shoot, and you have an image here of it forming new leaves, and the apical meristem is going to be at that tip of the shoot. It can also be here in the lateral bud, so if something were to happen and this would be removed, you would have new growth right here. And then we have the apical meristem associated with the root, and that's going to be here just above the root cap here. Apical meristems produce the primary plant body in 3 distinct populations: the protoderm, which is the dermal tissue, the ground meristem, which is the bulk of the plant body, and that's where photosynthesis takes place and storage happens, and the procambium, which is vascular tissue. And remember, that's primarily for transport, but also for support. Here are just some images of that, but it's cool to think about a cross-section of those things and how it would look if you're looking at a cross-section of a plant. Here you can see that we have dermal tissue on the outside, ground tissue in the inside, and here we have the vascular tissue in a sort of sun-like pattern. And then if we're in the root, we'll have dermal tissue once again on the outside, ground tissue is going to be right here, and then we're going to have a sort of a solid vascular tissue for transport of nutrients and water. So how is the primary root system organized? Well, here's an image that's from your book that shows you that procambium, the protoderm, and the ground meristem, and then the apical meristem just below that. But there's also the root cap, which is often confused with the area that the actual growth is happening. Remember, growth is happening here, so if you were to look at it under a microscope, you would want to focus on this area for mitosis to be happening. And then actually the cells right above it or below it, depending on where you're standing, those are going to be associated with the cap, or the root cap, and that will secrete a mucilage, which is a slimy substance, which helps lubricate the root tip so it can move through the soil more easily. And cells are constantly being replenished here, and this is also the area that senses gravity and determines direction of growth. So how is the primary root system organized? And where does growth happen? Well, we already talked a little bit about it. I mentioned that growth is actually happening right here. So if you were to take the tip of a root, as you might have done in 211 when we did cell biology, you would have actually not looked at the root cap for that mitosis to be happening, but you would rather look here at the apical meristem. That is where the growth is actually happening. So we divide it into actually 3 different sections. There's the zone of cell division, right? So I said mitosis happens here. The zone of cellular elongation, and then the zone of cellular maturation, which is where we have a lot of the root hairs and lateral root. Remember, the zone of maturation are going to be older cells. It makes sense, it uses the term maturation, and they're going to be differentiating into that dermal, vascular, and ground tissue. Including, don't forget that in the cell division, that's where we're going to have actual cell division happening. And then in the area of elongation, it's just what you think it is. It's actually where cells are actively increasing in length. Just to mention, I said in the zone of maturation we have root hairs. Remember that root hairs are going to be tiny little outgrowths of the root. They're going to comprise entirely of epidermal tissue. They increase surface area, so there's a large surface areas for absorption of water and nutrients, and they're the actual sites of water and nutrient absorption. And this is just showing you a cross-section of a monocot versus a eudicot. And a eudicot is our old term. You may have heard or remembered learning about dicots, but now we say eudicots, true dicots. And eudicots have this sort of much more circular here pattern where you have vascular tissue in a round pattern, while a cross-section of monocot system, you're going to see lots and lots of tiny little dots, sort of like a dartboard, and that's going to be your vascular bundles. And in between all of that is your ground tissue, which goes for the same here. And the difference here is we have differentiation of these cells where we talk about it on the outside of that vascular bundle as the cortex, and on the inside we call that the pith. And then both of them have an outer epidermis. So that's that organization we talked about, and I've already mentioned it here where I use those terms vascular bundle, and in the ground tissue we talk about pith and cortex. This is just showing you what you probably already remember about what a plant cell looks like. Don't forget that we're talking about cell walls here. We have a cell wall in a plant. We're going to have vacuoles, which are going to be unique to plants, and then we're going to have those chloroplasts. Mitochondria, remember, exist in plants as well as animal cells, so don't think that animals have mitochondria and plants have chloroplasts, because plants actually have mitochondria as well. And remember that you may have learned about something called the plasmodesma. And remember that is a connection between two different cells, and it allows for a cytoplasmic connection between the two of adjacent cells. And so those are some things that you should remember in terms of how we have the setup of that cell of the plant, where we talk about the cell wall, the plasmodesma, desmata, which is just plural of that, and chloroplasts and vacuoles. And remember, the vacuoles will contain cell sap as well as store wastewater and nutrients. In the dermal tissue, we're going to have the stomata, or a stoma is just a singular one of those. And remember, we talked about in plant evolution that we start to see this idea of the pore very early on as an adaptation, because if we have a cuticle and it's too dense and it doesn't allow for gas exchange, a plant could not survive. So what you have is pores developing already in those early mossin relatives. And then a true stomata is going to have guard cells on either side. And this is what a stoma looks like open, and this is what it looks like closed. Remember that guard cell is here and the pore is actually in between here. And here are just some examples of what that looks like. They always look like little tiny mouths to me. And here's just a couple of images of that dermal tissue, and this is on a lyrata leaf, and you can see that this is a type of Arabidopsis plant, and you see a single stomata here, or single stoma, and many stomata here. Dermal tissue is, remember, that interface, just like it is for us. It's the interface between the outside and the inside. And so we're going to have many different cell types with particular structure and function. So I talked about the waxy cuticle. Remember, that's associated with minimizing water loss. And it's also a defense against pathogens. And this is where the stomata are going to be located, and trichomes, which are hair-like appendages that are protective. So what are trichomes, and what are the function of trichomes? Trichomes are one of my personal favorites because they are so cool to look at and so incredibly variable. And they play a big role, particularly in defense against herbivory from insects. So it's obviously a favorite of mine. This is just an example of what they look like. And so they may not be very big. You could probably feel that structure very lightly with your fingers if you touch a plant. But for an insect, it's like a forest, right? So this is just showing you an image of what that insect is actually dealing with when you look it up close. So here's the tarsi, or the leg of that insect, and you can see it's like a little, like a high grassland, right? And they can also have urticating hairs, which means they secrete some sort of substance that bothers an organism. They can have toxins associated with it, so they perform an array of functions. This is showing you some of the diversity that we see in trichomes. They often give a fuzzy appearance, so when you see the fuzziness of a sundew, those are trichomes. And then you can also have them branched. This is the lyrata again. We can have these crazy branched ones that look like antlers, and then there's also multi-branched. They look like little stars, and these are all different types of basic defense for the leaf of the plant. So they're protective and hair-like, and they're specialized epidermal cells. And as I showed you, they come in a really, really great variety of different shapes and sizes. They are able to or help the leaf keep the surface cool by reflecting sunlight. They also reduce water loss because that dense mat can limit transpiration. As I told you before, can provide barbs, store toxic compounds to prevent herbivory, and they can also, in the case of things like our sundew plant, trap and digest insects. Ground tissue, the whole system there, remember, is most where photosynthesis takes place and carbohydrate storage. And this is associated with 3 distinct types of cells: the parenchyma, the collenchyma, and the sclerenchyma. So I already sort of mentioned what cells make up ground tissue, but what are the functions of each of these cells? This is a really nice diagram of all the ground tissue, including the vascular tissue, as you can see with the xylem and the phloem. It's nice because it gives you an image of it from the top of the leaf down to the bottom of the leaf, and a lot of the different important types of structures that we find, including things like the companion cells, the tracheids, the pits, the different types of ground tissue cells like the collenchyma, and things like the vessel element and the sieve tube element. So the parenchyma are the workhorse cells. You can see that in leaves, they are responsible for photosynthesis and gas exchange. And in the roots, they're going to be where that carbohydrate storage is. And you can see if you looked at them under a microscope, you would see those starch granules like in a potato. Parenchyma cells are relatively thin have relatively thin primary cell walls. They're abundant. They're the most abundant and versatile in plant cells. And as I mentioned already, in leaves, they're the primary site of photosynthesis, and in the roots, they're where we see the storage of starch. Most are totipotent, which means they can divide and develop into a mature plant. So that is the idea that if you have some kind of cutting, you can actually clone the plant that you have. So that makes them very, very good for propagating new plants. So you can cut a piece of the stem and place it in water. This is an example of that you can see here. And the parenchyma cells will divide to produce a callus, which is just undifferentiated cells. And then roots develop from the callus, and a new individual plant can arise from that cutting. A lot of different plants do that. Bananas and seedless grapes, for example, which are important because we use them in agriculture. Collenchyma cells function mainly in support. You can see the classic example, a celery stalk, and that stringy part is part of that structuring and support for the cell. We can see that here in a cross section of the collenchyma cells. They vary in thickness. They continue to lengthen even when they're mature, so they provide structural support, and they're abundant in elongating stems and in the petiole of leaves. Sclerenchyma cells are specialized for support. You'll hear the term fibers and sclerids associated with them. It is where we see that lignin that we've talked about when we talked about plant evolution. So these elongated fibers are associated with support, and the short sclerids are associated with protection. And we've already mentioned that that secondary wall contains lignin, which means it's unable to actually expand, but it's important for support. Ground tissue typically contains 2 types, those fibers and those sclerids that we talked about. And then last but not least, we have vascular tissue. This is probably what you're the most familiar with, the phloem and the xylem for transport of water and sugar and other nutrients. Remember, the xylem conducts water, and in that water we have dissolved ions. It's one directional from roots to shoots. The phloem, on the other hand, conducts sugar, amino acids, chemical signals, lots of different things, and it's in both directions. This is just some of the types of structures you'll see in the vascular tissue. We have tracheids. These are nice pictures of the tracheids here, the vessel elements, and how they work in tandem with each other. We're going to go way more into this when we talk about sugar and water transport, but you should just be aware of what types of cells they are and what kind of tissue they're located in. It is important also to remember that it's vascular plants that have tracheids, and so it's the xylem that will contain that. And in angiosperms, we have vessel elements. There are a few other species that have that. And in angiosperms, we have tracheids and vessel elements that are found adjacent to each other. Also remember that xylem cells are dead at maturity and they're filled with fluids they conduct instead of cytoplasm. The structure of tracheids and vessel elements are a bit different. Tracheids are longer, slender cells with tapered ends. They have pits where only a primary cell wall is present, and the water moves between tracheids through pits, while vessel elements are shorter and wider. They have pits and perforations, and their openings lack that primary and secondary wall, and they conduct water much more efficiently than tracheids. And this is a picture of the phloem structure. So the phloem, we're going to be talking about sieve tube members and companion cells. There is an image of that here. We see these two different types of cells and their companion cells next to it. And the sieve plate, which is shown here. And this is just looking at it from a longitudinal section. The sieve tube members are long, thin cells that lack nuclei, chloroplasts, and most other organelles because they're responsible for transporting. So they don't really need much because they're transporting sugar and nutrients. The companion cells are non-conducting, but they're very important in the unloading and loading of sugar. So we've talked now about primary growth, which is where we're lengthening the roots and the shoots and extending the plant's reach within the soil or within the air so that it can access potentially more light and acquire CO2, water, and ions. Now, secondary growth is going to be all about extending the width of the plant and providing structural support as the plant gets bigger. It produces wood, and we'll talk a little bit about cambium, which is that secondary meristem, as well as increasing the amount of conducting tissue. So we're going to start to talk about secondary xylem and phloem, which is, remember, only in gymnosperms and some angiosperms. So what's cambium and the types of cambium? Remember, cambium is just secondary meristem or lateral meristem. It's made up from thin layers of meristematic cells that form cylinders, and cells divide in a way that increase the width of the roots and shoots. And this is just showing you that image of it. Remember, we talked about the secondary phloem and the secondary xylem. We have cork cambium, which is going to be right here and increasing in this direction. And our growth is really going to be happening right here with the vascular cambium. So two types of cambium secondary growth. I mentioned it when we looked at this picture here. We have cork cambium and then we have vascular cambium. Vascular cambium forms between the secondary growth of vascular tissues. Remember that secondary phloem and secondary xylem. And cork cambium is located near the perimeter of the stem and produces new cells to the outside. So vascular cambium produces both phloem and xylem. Remember, phloem is going to be new cells produced outside of the meristem, and xylem, secondary xylem, is going to be new cells to the inside. These are only found in gymnosperms and some angiosperms. The secondary phloem is in sugar transport. We call that, when it's combined with cork cambium tissue, bark. And the secondary xylem, which is still water transport and structural support, and that's going to form wood. So how does bark form? This is just showing you an example of how that forms. And its role of it is really to protect woody stems and increase the girth, replaces epidermal tissue as a woody stem or root matures. It prevents water loss and also protects it against pathogens and herbivores. And there are sometimes adaptations associated with trying to increase gas exchange, even though we have this very strong protective layer. So we'll see things like lenticels, which are going to be in the bark, and they look sort of like lines in the bark, and they are spongy segments of bark for gas exchange. And a lot of native peoples, particularly here in the Pacific Northwest, have taken advantage of that and harvest bark to make things. And this is just a example here of harvesting of bark from a cedar, a western red cedar, and how this bark is harvested off of a dead tree and how it's made into something like a basket. So you may have heard the term heartwood. So heartwood is going to be associated with what is very, very much in the center. It's going to be where we see accumulation of protective compounds, so resins and gums. While on the other hand, we have sapwood, which is only the newest xylem tissue, and it continues to transport water. So the structure of the tree trunk. Remember that trees are long-lived perennials, so as the tree grows and matures, only the newest xylem tissue continues to transport water. And as I mentioned before, we're going to have the innermost xylem. It's going to have the resin and the gums. This is what causes it to darken. And then the heartwood is going to look like a darker colored inner xylem region, and the sapwood is going to look like a lighter colored outer xylem region. And this is some example of that. You see some of these darker and these lighter regions. And you can actually look at the structure of a tree trunk and know a lot about what that tree has experienced. This is a very interesting link here that looks at how we use trees to sort of understand how the climate's been changing. But as you can see, if we have a rainy season, we're going to have a very long white line here. If there's some sort of forest fire or some event happens, there's going to be sort of a gash in there. Dry seasons are going to be very small. And then we're going to have late summer, fall growth with these dark lines, and spring, early summer growth, which is the wide area here. And this is just an example of how you can take a tree and actually read the lines within the tree and know a lot about what the tree experienced, all the way from the early growth of the tree to maybe being shaded by other trees to even things like invasion by something like a gypsy moth caterpillar. Variation in the cell size results in annual growth rings. Periods of rapid growth are going to have large, secondary xylem cells are going to be large and thin-walled. Periods of dormancy, secondary xylem cells will be small and thick-walled. Wet conditions will have wide tree rings because the tree grows fast. Drought conditions will have narrow tree rings because the tree is growing slower. This can also be years when there is abundant acid rain, air pollution, or any kind of thing that might challenge the growth of the tree. Thank you so much for watching.